If you want, today will be without break lecture, yes, and we'll finish more early, yes. It's better for you. Yes, at first I want to. Pardon, okay? So first of all, I want to show micrographs, yes, which are included in the quiz. We will repeat this part, then we'll start to talk about the other organs of the digestive tract. Okay. Da sunt de meciros feliz. Okay. So, I hope you know all these micrographs, all these structures, but also you have to know the functions, features of these structures. So, what's this? It's filiform papillae. What is important for this type of papillae? So this type of papillae is the most numerous, yes, without taste buds, yes, and the heavily keratinized they are. The next one, it's the same, yeah, it's the same type of papillae. It's filiform papillae again. And what about this one? Folate, yes. So what you know about this type of papillae? It's on both sides, yes, of the tongue. Its rudimentary structure is more developed in children, yes, than in adult individuals. Are taste buds on the surface of such type of papillae or not? Yes, they are visible. Yes, here. This. Okay. What about this? It's volate, circumvolate papillae. How many circumvolate papillae are located on the surface of tongue? 8 to 12, yes, 8 to 12. Yes, and many taste buds are. Yes, here. And these? Fungiform, yes, fungiform, yes, fungiform. Also, some taste buds are located on the surface of the fungiform papillae. And what is indicated now in this micrograph? Taste bud, yes, it's taste bud. And these structures, the taste buds, which type of cells are in taste buds? How many types of cells and what's the function of these cells? 3 different types of cells. Yeah. So the 5,100 cells compose the taste bud, and they are 3 different types. Especially 2 are the main types of cells: gustatory cells and supportive cells, yes, and also are basal cells. Of the what's the function of the basal cells? They are stem cells, yes. They give rise to other type of cells. The both type, more correct, are sensory cells and sensory gustatory cells and supportive cells. Supportive cells, they are immature cells, yes, here. Okay, so what's this? Mucous gland, yes, mucous gland. Mucous glands are small mucous glands in the oral cavity. Yes, and this, it's the same, it's the mucous gland, yes. This one, serous, yes, it's serous gland. And what's this? Yes, it's the cross section. Do you know the layers of these? What are these layers, the main layers? Yes, mucosa, submucosa, muscularis, and Adventitia, yes. For esophagus, the outer layer is adventitia, yes, of course. Which type of epithelium? Stratified squamous, non-keratinized, yes? Okay. And what about this? Yes, it's again esophagus. Yes, stratified squamous epithelium, then lamina propria, muscularis mucosa, these three sublayers for mucosa. Then is submucosa. Something specific is for esophagus in submucosa. Yes, glands. Yes, the mucous glands. Then these are mucous glands. Are visible these blood vessels here and ducts? Yes. And this one. Yes, it's the same. It's the longitudinal. Yes, this section. Here, another layer also is visible, muscularis externa, and part of adventitia with adipose tissue here. And what about this? This is stomach. Yes, it's stomach, of course. And this is surface of stomach here with pits. Invaginations are visible here. The epithelial lining forms invagination, the pits. And what are these? They are pits. Pits are this. Look here, they are pits. And they are gastric glands. Yes, long glands are in stomach. Yes, they are glands. How you think, which part of the stomach is this? Body or fundus? Why? Yes, here parietal cells. Yes, visible, yes. And what's this? Rogae of the stomach, yes, it's the mucosa with submucosa, it's rogae, and the pits are visible and glands. Yes, here. Okay, and this? Again, stomach, yes, stomach, mucosa with glands. It's also the large part of the stomach, yes, large part of the stomach, because they are parietal cells and chief cells. More darker stained chief cells also are visible. Do you remember the enteroendocrine cells of the stomach glands? Yes, and they are open or closed type enteroendocrine cells in stomach. Closed, yes. In intestine we have the open type enteroendocrine cells, yes. So now about this part. Yes, it's duodenum. Why this is duodenum, not jejunum? Yes, Brunner's glands in submucosa. Yes, the mucous glands are here. And this? It's the same, it's the part, yes, of the duodenum. And which type of epithelium covers the surface of the of intestine? Yes. Simple columnar epithelium. And the what are the main cells of this epithelial lining? Enterocytes and goblet cells, yes. So in a large intestine, are goblet cells or not? Yes, and the most numerous they are, yes, compared to the small intestine. Yes, of course. And what's this cell? What is this cell, and what's the function of this cell? Yes, it's Paneth cell located more lower part of the gland. Yes, the Paneth cell with large acidophilic granules. And what's the function? Yes. Okay, now what is this part, this segment of the small intestine? It's jejunum, yes. Great. And what about this? Ileum. This? Ileum, yes. Lymphatic tissue is visible, yes, in the center. M cells. Do you remember M cells of lymphatic tissue? Yes. And do you know what is this? Yes, appendix, yes. This is the lymphatic tissue. Appendix, which is the secondary lymphatic organ. Yes. This way, about the other micrographs, they are for the next quiz. So all micrographs, I think, you know. Yeah, I showed these micrographs during tutorial, and tomorrow we also have tutorial, yes? In histology, okay. So micrographs, I don't remember. Not many. But the micrographs, you all know these micrographs. Today we are starting to talk about the very important organs. So we know digestive tract, and we have started to talk about the organs which are associated with the digestive tract. Yes, we know salivary glands. Yes. What are the major salivary glands? Yes, and many small glands, yeah, mucous glands throughout the oral cavity, you know all this. And today we have to talk about liver and gallbladder. And yes, also, you know, pancreas, yes, such as the exocrine organ, the pancreas. Okay, now the liver, which is the largest organ of the body, and its weight's about 1.5 kilogram. It's the 2% of the total weight of the body. And the liver is the really very important organ which has many functions. And the liver has 2 large lobes and 2 small lobes. So what are the large lobes of the liver? It's the right lobe and the left lobe. And 2 small lobes also are here, the caudate lobe and quadrate lobe. 2 large and 2 small lobes. And liver is covered by capsule, yes, by thin connective tissue capsule and mesothelium, part of the visceral peritoneum. And so then septa divides into such lobes, large and small lobes, yes. Liver is really important because it has endocrine functions, exocrine functions. The liver is the parts, it's the structure between the blood and the digestive system, yes. And so what are the now functions of the liver? Secretion of course, this function, you know, secretion of bile components. This is the exocrine function of the liver, secretion bile components. Then, synthesis and endocrine secretion into the blood of the major plasma proteins. This is endocrine function of the liver. Then, gluconeogenesis. This is conversion of amino acids into glucose. Detoxification, it means that the breakdown and the conjugation of ingested toxins. Amino acid deamination, producing urea removed from blood in kidneys, then storage of glucose in glycogen granules and triglycerides, storage of vitamin A, but this is so about the cells. Okay, I'll talk more later. And so the storage of vitamin A and other fat-soluble vitamins, removal of effete erythrocytes by specialized macrophages or Kupffer cells, storage of iron in complexes with the protein ferritin. So you can see so many functions. Yes, so many functions. Liver. Including such important exocrine and endocrine functions. So the main cells, the structural unit of the liver, what is it? It's the hepatocyte. Yes, the liver cell, the main cell is hepatocyte, and these hepatocytes, they are large cells with round nuclei. They have usually one nucleus, maybe sometimes the two nucleus, also large cells, hepatocytes. These hepatocytes form plate, and such 100 such plates they form, and these plates are located radially around the vein, central vein. So this is the branch of the central vein, and such thousands of such lobules form the tissue of the liver. So liver tissue is a very specific tissue. It's very easy to understand that is tissue of the liver because it has such specific lobuled structure. So I said liver is covered by connective tissue. Yes, the capsule, and this is thin capsule, but more thicker is in area where enters and exits blood vessels, where exit lymph vessel and enters nerve fibers, and so which arteries enters into the liver and the veins? Hepatic artery, hepatic vein, yes, portal vein are the arteries and the veins of the liver. So the cell, liver cells, hepatocyte, yes, hepatocyte, and I'll show then ultrastructure of the cell. Hepatocytes form plates, and these plates then form the lobule. Yes, you can see, yes. So the arrangement of these plates located around the central vein. Okay, now structure, yes? Plates located around the vein. This is one lobule, liver lobule, yes, liver lobule. This is branch of central vein. This is central vein here. And between these plates, between hepatocytes, run hepatic sinusoids. So this is central vein. Yes, around central vein you can see plates composed by these hepatocytes. And now what is periphery here? At periphery are portal triads. 3, 4, 5 such portal triads are, and triad, it means 3 different components. Yes, components, 3 different components. What are these components now? Branch of bile duct, you can see these. Branch of hepatic portal vein, and branch of hepatic artery. So branch of hepatic artery, branch of portal vein, and branch of bile duct form portal triad. And these portal triads are located at the periphery of the lobules. Yes, sometimes they also contain lymph vessels. Here lymph vessels also are sometimes at the periphery. Okay, now I want to show this tissue, the portal triad. Yes, it's the hepatic lobule. Yes, it's hepatic lobule. In the center, here is the central vein. Yes, hepatocytes are located around the central vein, and this is the portal triad. What are the parts of the portal triad? What is this? Hepatic artery. Yes, it's a hepatic artery. Yes, small hepatic artery is here. Then what is this? Portal vein, branch of portal vein, bile ductal here, and lymph vessel. Yes, lymph vessel. So connective tissue covers the liver, and liver does not contain many connective tissue, but most of the areas where is connective tissue are the portal triads. So usually connective tissue is located in portal triad. Here is these are the sites where is the connective tissue. Then it's also, yes, the hepatic lobule, but this is hepatic lobule of the pig, and this is hepatic lobule of the human. So can you distinguish any difference between these two? This is, yes, it's the hepatic lobule of pig. Another one is the hepatic lobule of human. What's the difference, how you think, between these two? Yes? Lobule is more visible, yes, here. And boundaries more visible. So around this lobule are reticular fibers, usually are reticular fibers, but in some species, for example in pigs, they are more developed compared to the humans. Yes, these boundaries and such the lobules are more difficult to distinguish the lobule to each other, unlike the pigs. In pigs, each lobules are more visible. This is the one lobule, another lobule, and here is more difficult to find such lines. And portal triads, yes, the portal triads, here also is visible portal triad. What are these? Are the ducts here, artery, vein, yes, this bile ductal, vein, artery. So hepatic triads. It's clear, yes, what does it mean, the hepatic triad? Yes, located at periphery they are, and they contain the branch of artery, branch of vein, and bile ductal. So liver is the organ, yes, between the digestive tract and blood, and liver receives blood two different sources. Yes, the artery and vein. Vein, 75% of blood liver receives from the vein, and this is nutrient-rich blood but poor in oxygen. And 25% of blood comes from the artery, and this is oxygen-rich blood but nutrient-poor blood. So 75% nutrient-rich blood receives the liver, yes, and this blood is from the stomach, from the spleen, yes, and 25% of blood is oxygen-rich blood. Here you can see the hepatic lobule microvasculature. Yes, it's the microvasculature. It means the small blood vessels and capillaries. Which type of capillaries are in the liver? In the liver, yes, between the plates of hepatocytes are sinusoids. So sinusoids, you know, yes, they are the type of the capillaries with large fenestrations are endothelial cells, and the with discontinuous basement membrane. So they are sinusoids. Okay, are visible hepatocytes here? Yes, these cells, large cells, acidophilic cells. Why they are acidophilic? They contain many mitochondria. Yes, and sinusoids are between these hepatocytes. And now here are the fibers. Reticulin fibers in liver are many reticulin fibers. So what does it mean, reticulin fibers? They contain the third type of collagen. Yes. Third type of collagen, reticulin fibers, and hepatocytes with round nuclei are hepatocytes, yes, with a large such round nuclei. This is, which part of the lobule is this? Look carefully, which part of the lobule? It's central part. Yes, it's the central here. The central. This is the central vein. It's central vein. Around the central vein are located the plates of hepatocytes, and on another micrograph, this is peripheral zone. Yes, it's peripheral zone here. Hepatic artery, portal vein, and bile ductal. How we can, do you know how we can distinguish? This is, for example, the artery, or this is duct? Vein is more larger, yes? But artery and vein, so the wall, they have the same thickness. Yes, the wall they have. How we can distinguish? Yes, please. Cells inside. So in vein is clear. Yes, in veins we have the large lumen, thin wall, and inside lumen are red blood cells usually. Yes. If more similar are the veins and lymph vessels, and arteries and ducts. How we can distinguish? This is lymph vessel or this is vein. The first question is this, and another question is the artery and duct. Have you any idea? Vein, or this is lymph vessel, or this is vein? Yes. Lymph vessel or vein. In vein, yes, in lumen are red blood cells. Not in lymph vessels. Yes, lymph vessels are without. If are in lymph vessels any cells, they are not red blood cells. They are not erythrocytes. Usually they are erythrocytes, so they are lymphocytes. Yes, lymphocytes. So not red blood cells. But this is artery or duct. Now, so in ducts, yes, the wall of the duct. Usually in ducts are cuboidal or columnar cells. Yes, ducts wall are cuboidal or columnar cells. In arteries, the inner layer is are endothelial cells. Endothelial cells, they are not cuboidal, they are not columnar. Yes, they are squamous cells. They have elongated nuclei, not the round nuclei. In ducts nuclear cells have round nuclear, not in ducts. In ducts, in inner layer cells, they have elongated thin nuclear. Is it understandable? Okay. So this is the central part, it's the central vein, and are visible sinusoids, and this is the portal area, and the connective tissue is visible. Yes, the collagen fibers are stained red color. Okay. Now the direction of the blood. Blood direction is from the periphery to the center. Yes, this is direction of the blood from the periphery to the center. This is the portal area. Okay. This is now the ultrastructure of hepatocytes. So ultrastructure of hepatocytes. You can see so many organelles. So many organelles are located into the cytoplasm of hepatocytes. Yes, and part of nuclei, of course, they have euchromatic nuclei because they are really very active cells. Then numerous mitochondria, yes, numerous mitochondria are visible here. Well developed both type of endoplasmic reticulum, rough endoplasmic reticulum. Yes, it's the rough endoplasmic reticulum. Here proceeds the synthesis, of course, protein synthesis. Also, you can see the part of the smooth endoplasmic reticulum. Detoxification proceeds, yes, in these cells. Then, Golgi, yes, the Golgi complexes also are here. Glycogen granules. Lysosomes also are peroxisomes. Yes, the peroxisomes also are. So what is the function of peroxisomes? Do you remember these organelles, peroxisomes? Yes, what's the function of peroxisomes? They contain different enzymes, yes, for oxidation, catalase, and they are important organelles for detoxification. Okay, and this is the sinusoidal space, and this is perisinusoidal space, and these cells have projections into the perisinusoidal space. So these cells, hepatocytes, they have very close contact with blood. Yes, with blood, and these projections enter into the sinusoidal space. This is endothelial cell here, and look here at this part. Starts bile canaliculi, small bile duct. Yes, and small bile duct here. Okay, so perisinusoidal space, this one is the perisinusoidal space, and another one is the bile canaliculi. You can see three-dimensional micrographs here, and the perisinusoidal space and endothelial cells. You can see, yes, fenestrated endothelial cells, and many fenestrations are visible on the surface of these. So, and they are stellate microphages, usually called Kupffer cells, and hepatic stellate cells or Ito cells. They are located into the sinusoidal areas. Yes, into the sinusoidal areas they are located. Kupffer cells. So they are microphages, and they are formed from blood cells. Do you know what are the cells? The cells, precursor cells of the Kupffer cells. Kupffer cells. Removal of debris, yes, removal of the debris, removal of damaged, yes, the damaged cells, the phagocytic function they have, and they are Kupffer cells. As this is it's the nuclear of Kupffer cells, and they have well stained, yes, dark stained, elongated nuclear, and it's the nuclear of hepatocyte, and this is the nuclear of Kupffer cells. It's very easy, yes, to distinguish which one is the hepatocyte and Kupffer cells. They are acidophilic, and with the light stained and round nuclear hepatocytes, Kupffer cells, and are located in perisinusoidal, such the parts, the places, and they have darker stained elongated nuclear. And Ito cells. So Ito cells, I want to show, these are Ito cells. They also are located into the sinusoidal places. Yes, these Ito cells. They contain lipid droplets, yes, the function vitamin A storage and other fat-soluble vitamins storage, and lipid droplets. Can you see these lipid droplets? Yes, this is, yes, it's the Ito cells. It's Kupffers. So I'll show all these type of cells. These hepatocyte, yes, have a large cell, then Kupffer cell, and this is the Ito cell here. Endothelial cell, it's the perisinusoidal space. Yes, sinusoids are here. Okay, now we know, yes, the functions of all these types of cells. Now I want to show damaged tissues of the liver. Fatty liver disease is a reversible condition in which large lipid droplets containing triglycerides accumulate abnormally in hepatocytes via the process called steatosis. And this disorder has multiple causes, but it occurs most commonly in individuals with alcoholism or obesity. Accumulation of fat in hepatocytes may produce a progressive inflammation of the liver on hepatitis, in this case called steatohepatitis. This is tissue of normal liver, and these are tissues of damaged liver. This is fatty liver disease, and you can see yes, the adipocytes, yes, adipocytes between the cells of the liver, and this also is the damaged. And you can compare, yes, you can compare the color. This is the healthy liver, normal liver, and the damaged liver. Yes, it's this liver has more yellow color, and lipids, yes, lipids give such color. Then, yes. This? So, two main reasons. Yes, the two main reasons may be here, alcoholism or obesity. It's the reason of obesity. Yes. Okay. So, most malignant tumors of the liver derive from hepatocytes or cholangiocytes of the hepatic ducts. So, the cholangiocytes are cells of the ducts. So, not only alcohol is, yes, the reason of the damages, Okay, so the pathogenesis of liver carcinoma is associated with a variety of square disorders such as chronic viral hepatitis. Of course, you have heard this hepatitis B or C, yes? B or C, and cirrhosis, liver cirrhosis, of course, you know, and the cancer, liver cancer, especially nowadays, are often cases. So this, yes, the damaged liver, and the histological section of the liver tissue, liver carcinoma. Yes. Cholangiocytes, what are? Hepatocytes or cholangiocytes. Yes. Ah, yeah. So the cholangiocytes are the ducts. Yes, the duct cells of the liver ducts. Both leads the very serious problems. Yes. So the liver, the tumor of the hepatocytes, it leads to then to cholangiocytes. It leads to the damages of the ducts than the liver, but they both are very sensitive cells. So they usually such tumors arises from epithelial cells. Yes, epithelial cells are the most sensitive for such tumors, and liver cells are epithelial cells. Okay, then okay, so the next yes, now the normal liver and liver cirrhosis. For cirrhosis, one of the main reason is alcohol. Yes. Now, in the normal liver, most dense connective tissue is found only in the portal areas surrounding the blood vessels and bile ductal. In liver cirrhosis, which occurs late in chronic liver disease, fibrosis, and the proliferation of fibroblasts and hepatic stellate cells occur beyond the portal areas. The excessive connective tissue may disrupt the normal hepatic architecture and interfere with liver function. So when proceeds the damages, yes, of these tissues forms the fibers, yes, the fibers, and forms the fibrosis here, and forms the connective tissue, and if is the large amount connective tissue, of course it means that the liver is damaged. Yes, liver is damaged. If you remember in heart, after the damages, yes, of the heart, forms the connective tissue. Yes, we are in places we are the damaged cells. The same, yes, here, if it's the more connective tissue as in normal condition, it means that this liver is damaged. And I want to show again the normal liver and tissue of the normal liver and the fatty liver. Yes, the fatty liver and the cirrhotic liver. So what's the difference? Yes, between fatty liver and cirrhotic. Yes, the liver. Normal liver, you can see the architecture of so these hepatocytes without such lipids, without fat cells are, and the fatty liver, tissue of the fatty liver, and you can see so many, yes, the lipids are here, the fat cells are here, and where is the cirrhotic liver? Not fat cells, but are the tissue is damaged, and the fibroblasts are here. Yes, between the hepatocytes it forms the connective tissue, and in both cases, the liver, so view of the liver is changed. Yes, the tissue of the liver. So it's hepatocyte. This is the ultrastructure of hepatocytes. About this we talked, but again I want to talk again about this because it's more visible. As here the structures and also the functions of the liver. So in the center, round nuclear. Yes, the round nuclear, euchromatic, of course, the nuclear, and in this, yes, the here are endothelial cells. So endothelial cells are visible, and reticular fibers. So fenestrations also are visible. Now, what inside these, inside hepatocyte, which structures are? First of all, mitochondria, of course, many mitochondria, rough endoplasmic reticulum well developed, then smooth endoplasmic reticulum also, then Golgi apparatus, yes, lysosomes, lipid droplets, then also peroxisomes, yes, and glycogen granules. In liver, lipofuscin granules also are. In which cells are lipofuscin granules? Do you remember the cells where are typical such granules? In nerve cells, neurons. Yes, are lipofuscin granules. In myocardial cells, long-living cells. Yes, long-living cells contain lipofuscin granules. Okay, so, and this is perisinusoidal space. Perisinusoidal space is here, but on this lateral side starts bile canaliculus. Here you can see desmosomes and different type of junctions, tight junctions, desmosomes are visible, and starts by such small bile canaliculae. Yes, bile canaliculae, okay, bile ductals. And bile ductals, yes, they at first are very small bile ductals, then forms more larger canals, and finally forms the really large bile duct. Yes, and these bile ducts are lined by cuboidal epithelial cells, and these cells have such name. They are cholangiocytes. Yes, cholangiocytes. Then, now I want to talk about these structures. These are, so these structures show the functions of liver. So, structural unit. Yes, the structural unit is hepatocyte. Yes, but functional units are lobules, and three different such ways shows the functions of the liver, starting from the first one. The first one is the classic hepatic lobule, and it shows that the blood direction and blood from the periphery, blood from the portal vein and the hepatic artery, its blood runs to the central vein, to the hepatic. Yes, on the central vein, from the periphery to the center. This is classic, it's the classic hepatic lobule. Now another one, portal lobule. So what does it show? Portal lobule. It shows the bile direction, and the portal lobule is formed. So this formed, and the three such lobules together, forms and shows the portal lobule. And so in this center is the peripheral zone of lobule, is the portal triad, and it's and this is the direction of the bile. Bile runs opposite of the blood. If blood from the periphery to the center, bile opposite from the center to the periphery. Yes, and from three such lobules, bile moves to this direction and enters to this bile ductal. And the third one, hepatic acinus. What now about hepatic acinus? What does it mean? You can see two lobules. In the center, it's the peripheral zone. It's peripheral zone here in the center, and these cells, hepatocytes, they are part of the hepatocytes, of course. Parts are located into this near the central vein, and other hepatocytes are located more peripheral. Yes, more into the peripheral zone, and they differ by internal organization, by the organelles, and also they differ the function. Why? Because these cells which are located near the portal triad, they receive oxygen and nutrient rich blood. Yes, oxygen rich blood by the artery and nutrient rich blood by the vein. So they are the cells which are the first zone cells which receive rich blood, oxygen and nutrient rich blood. And in these hepatocytes proceeds active synthesis. So the hepatocytes which are located near the central vein, they receive more poor blood. Yes, they are the third zone hepatocytes, and their function is more detoxification than synthesis. So, and between this is the second zone. They are located in middle zone. Yes, the second zone. So these cells located here, the zone is the first zone. They receive more oxygenated, and the third zone least oxygenated blood, and the first zone of course receive more nutrient than the hepatocytes of the third zone. So is it also, yes, understandable what's the difference between these hepatocytes and its influence on their structural organization? Yes, and more peroxisomes contain the hepatocytes which are located into the third zone, and well developed is the rough endoplasmic reticulum, especially of the cells which are located into the first zone. If this is work, I want to show this short video, but this is without voice, but anyway, yes, all this you know. So the liver, and you can see what is this. It's gallbladder. Yes, here it's gallbladder, it's liver, and this is the site where enters and exits blood vessels and bile duct. So portal vein carries blood from spleen, pancreas, intestines, and 75% of blood carries portal vein. Hepatic artery, 25% of blood liver receives from hepatic artery, yes, and hepatic artery carries oxygen-rich blood. Yes, in the center is the hepatic vein, and the branches are central into the center in each lobule are the branches of the central vein. So, bile secretion is the exocrine function of the liver. Yes. This is the lobule. It's the lobule. And you can see periphery of the lobule with portal triad. So in the center is the central vein, of course. Yes, it's the peripheral zone, portal triad, the branch of portal vein, branch of hepatic artery, and the bile duct. Or sometimes lymph vessels also are here, yes? So 3, 4, 5, 6 such portal triads are located at the periphery of each lobule. So into the central vein, yes, and the periphery portal triads. So classic lobule. What does it mean, classic lobule? It shows the blood direction. Yes, the blood direction. Yes, blood runs from the periphery to the center. And what is this? It's the direction of the bile, yes? And liver acinus with 3 zones, yes, with 3 zones. So from one central vein to another central vein, yes, is the distance. And now we can see the hepatocytes of different zones. So the first zone, they are the first zones, hepatocytes, which receive rich blood, oxygen and nutrient-rich blood, and the main function of these hepatocytes is the synthesis. And the third zone, hepatocytes, yes, they receive the poor blood, and they more are involved in detoxification. Sometimes classic lobule is named such as anatomical unit, and the liver acinus the functional. Yes, the functional unit. Okay. So This part is understandable. Yes, the functions of the hepatocytes. Okay, now the next structure is the gallbladder, and you can see here the biliary tract also, and left and right hepatic ducts join and merge to form a common hepatic duct and common hepatic, and the cystic duct merge to form a common bile duct. Yes, the common bile duct, and main pancreatic duct merges with common bile duct at the hepatopancreatic ampulla, which extends into the duodenum, and bile and pancreatic juices enter duodenum at the major duodenal papilla. So what is this? This is the gallbladder. It's the gallbladder, and now I want to show the cells and the wall of the gallbladder. So this is the wall of the gallbladder when gallbladder is empty. Yes, empty, because you can see the folds of the gallbladder wall. Yes, it's gallbladder wall, and epithelial lining. These cells are simple columnar epithelial cells. Low columnar epithelial cells are here. Lamina propria. Yes, lamina propria. So gallbladder wall, mucosa. Then the submucosa is not developed here. It's not well developed submucosa, but well developed is muscularis, and you can see the adventitia. Yes, also in some parts is serosa, but most of the part is covered by adventitia. So mucosa, muscularis, adventitia are the main layers of the wall of the gallbladder. Epithelial lining, simple columnar epithelium, lamina propria, then is the muscularis, you can see, and the adventitia. This is the it's the cell of gallbladder. So what is important here? These cells have a few microvilli on the surface. Yes, on the surface a few microvilli they have, but have you seen these spaces? Spaces inside the cytoplasm, all these cells. So gallbladder. So what is the function of the gallbladder? Storage of bile. Yes, storage of the bile, and also proceeds here the concentration of the bile, and proceeds the water removal, and so the usually cells in which proceeds the active transports of the water, of the ions of the water, show such spaces inside the cytoplasm. Yes, inside cytoplasm. So glycogen granules are visible, numerous lysosomes also here, and so this gallbladder, this cell is the typical cell where proceeds active transportation of the water, of the liquid. And so what influence on the gallbladder activity? Cholecystokinin, yes, cholecystokinin. Where proceeds formation of the cholecystokinin? In small intestine, yes, in small intestine. Yes. Now, reabsorption of the water from bile in the gallbladder is involved in the formation of gallstones in the lumen of the gallbladder or biliary ducts, a condition called cholelithiasis. And this disorder usually originates with bile that already contains excessive amounts of normal bile components. And supersaturation of cholesterol in bile can lead to the formation of cholesterol stones, the most common form. And brown or black pigment stones can form when bile contains excessive amounts of unconjugated bilirubin, which can result from chronic hemolysis associated with disorders such as sickle cell anemia. And gallstones can lead to biliary obstruction, or more commonly to inflammation in acute or chronic cholecystitis. So, and okay, yes, these gallstones you can see here. Have you ever seen these stones? Gallstones? No? So it's really are large stones, the typical stones with such this smooth surface. Yes, the gallstones, really stones they are. And this is the picture of acute or chronic cholecystitis. Yes, you can see, so damaged is the wall of the gallbladder. And so of course the main function. Yes, here main functions have the liver and the gallbladder. In gallbladder only proceeds the storage, yes, of the bile, and sometimes it's necessary, yes, the surgical removal of the gallbladder when proceeds the inflammation of the gallbladder, and are the numerous such stones. And this after this surgical removal, the duct, liver duct and the bile duct is connected to the duodenum. Yes, directly without the gallbladder. And liver has many functions, and also liver has very important feature. Liver has regenerate. Yes, it has such function. It can liver tissue, and transplantation of the part of the liver from donor to the recipient, and the remaining part of the liver, this part can regenerate, and forms the liver, normally functioning liver. And so it's very important function. But in this case, what makes the difficulties? Yes, immune system, of course. Yes, in this. So after the transplantation, so the most important in this case then is the immune system, because this liver, it's the tissue after the damages. So after the damages, when is important, when it's necessary. Yes, the transplantation of the liver from one individual. Usually they are relations. Yes, maybe the mother or brother, sister. So and so when these donors and the recipients, yes, they can restore, yes, the liver. But for recipients, the important is the immune system. For immune system, this transplant, yes, this liver is the foreign tissue and starts the active reaction of the immune system. In this case, it's necessary to suppress the immune system, and the suppression immune system leads to another so the also factors. So any bacteria, any infections, it's the very harmful. Okay, but if liver has such feature, yes, such feature, regenerative feature, it's very of course it's very useful for the same organism. Yes, after the damages, when is necessary the surgical removal, some part of the liver can restore, and then after this the liver functions normally. Okay, so about liver, have you any questions? Sorry. No questions, it's understandable. And about the gallbladder wall, I think also I talked to what are the important features of this gallbladder cell. Okay, I think you want to finish, yes, and to have the time before quiz. Okay, thank you, and meet you tomorrow in the morning. Yeah, okay. And tomorrow I'll show micrographs about these organs.